Renin, also known as an angiotensinogenase, is an enzyme that participates in the body's renin-angiotensin system, also known as the renin-angiotensin-aldosterone axis, that mediates extracellular volume and arterial vasoconstriction. Thus, it regulates the body's mean arterial blood pressure. Renin is often improperly referred to as a hormone, although it has no peripheral receptors and rather has an enzymatic activity with which it hydrolyzes angiotensinogen to angiotensin I. Biochemistry and physiology. Structure. The primary structure of renin precursor consists of 406 amino acids with a pre and a pro segment carrying 20 and 46 amino acids, respectively. Mature renin contains 340 amino acids and has a mass of 37 kDa. Secretion. The enzyme renin is secreted by the afferent arterioles of the kidney from specialized cells called granular cells of the juxtaglomerular apparatus in response to three stimuli: a decrease in arterial blood pressure as detected by baroreceptors. This is the most direct causal link between blood pressure and renin secretion. A decrease in sodium chloride levels in the ultrafiltrate of the nephron. This flow is measured by the macula densa of the juxtaglomerular apparatus. Sympathetic nervous system activity, which also controls blood pressure, acting through the beta-1 adrenergic receptors. Human renin is secreted by at least two cellular pathways, a constitutive pathway for the secretion of prorenin and a regulated pathway for the secretion of mature renin. Renin-angiotensin system. The renin enzyme circulates in the bloodstream and breaks down angiotensinogen secreted from the liver into the peptide angiotensin I. Angiotensin I is further cleaved in the lungs by endothelial-bound angiotensin-converting enzyme into angiotensin II, the most vasoactive peptide. Angiotensin II is a potent constrictor of all blood vessels. It acts on the smooth muscle and, therefore, raises the resistance posed by these arteries to the heart. The heart, trying to overcome this increase in its load, works more vigorously, causing the blood pressure to rise. Angiotensin II also acts on the adrenal glands and releases aldosterone, which stimulates the epithelial cells in the distal tubule and collecting ducts of the kidneys to increase reabsorption of sodium, exchanging with potassium to maintain electrochemical neutrality, and water, leading to raised blood volume and raised blood pressure. The RAS also acts on the CNS to increase water intake by stimulating thirst, as well as conserving blood volume, by reducing urinary loss through the secretion of vasopressin from the posterior pituitary gland. The normal concentration of renin in adult human plasma is 1.98-24.6 ng in the upright position. Function. Renin activates the renin-angiotensin system by cleaving angiotensinogen, produced by the liver, to yield angiotensin I, which is further converted into angiotensin II by ACE, the angiotensin-converting enzyme primarily within the capillaries of the lungs. Angiotensin II then constricts blood vessels, increases the secretion of ADH and aldosterone, and stimulates the hypothalamus to activate the thirst reflex, each leading to an increase in blood pressure. Renin's primary function is therefore to eventually cause an increase in blood pressure, leading to restoration of perfusion pressure in the kidneys. Renin is secreted from juxtaglomerular kidney cells, which sense changes in renal perfusion pressure, via stretch receptors in the vascular walls. The juxtaglomerular cells are also stimulated to release renin by signaling from the macula densa. The macula densa sense changes in volume delivery to the distal tubule, and responds to a drop in tubular volume by stimulating renin release in the juxtaglomerular cells. Together, the macula densa and juxtaglomerular cells comprise the juxtaglomerular complex. Renin secretion is also stimulated by sympathetic nervous stimulation, mainly through beta-1 adrenoceptor activation. Renin can bind to ATP6/APRIL, which results in a fourfold increase in the conversion of angiotensinogen to angiotensin I over that shown by soluble renin. In addition, renin binding results in phosphorylation of serine and tyrosine residues of ATP6/APRIL. The level of renin mRNA appears to be modulated by the binding of HADHB, HUA and CP1 to a regulatory region in the 3'UTR. Genetics. The gene for renin, REE-nin, spans 12 kilobytes of DNA and contains 8 introns. It produces several mRNA that encode different REE-nin isoforms. Model organisms have been used in the study of REE-nin function. A knockout mouse line, called renin-1C enhancer KO, was generated. Male and female animals underwent a standardized phenotypic screen to determine the effects of deletion. 24 tests were carried out on mutant mice and 2 significant abnormalities were observed. Homozygous mutant animals had a decreased heart rate and an increased susceptibility to bacterial infection. A more detailed analysis of this line indicated plasma creatinine was also increased and males had lower mean arterial pressure than controls. Clinical applications. An overactive renin-angiotensin system leads to vasoconstriction and retention of sodium and water. These effects led to hypertension. Therefore, renin inhibitors can be used for the treatment of hypertension. This is measured by the plasma renin activity. In current medical practice, the renin-angiotensin-aldosterone system's overactivity is more commonly reduced using either ACE inhibitors or angiotensin II receptor blockers rather than a direct oral renin inhibitor. ACE inhibitors or ARBs are also part of the standard treatment after a heart attack. The differential diagnosis of kidney cancer in a young patient with hypertension includes juxtaglomerular cell tumor, Wilms tumor, and renal cell carcinoma, all of which may produce renin. Measurement. Renin is usually measured as the plasma renin activity. PRA is measured specially in case of certain diseases that present with hypertension or hypotension. PRA is also raised in certain tumors. A PRA measurement may be compared to a plasma aldosterone concentration as a PAC-PRA ratio. Discovery. Renin was discovered, characterized, and named in 1898 by Robert Tigerstedt, professor of physiology, and his student, Per Bergman, at the Karolinska Institute in Stockholm. See also, angiotensin-converting enzyme, plasma renin activity, renin inhibitor, renin stability regulatory element, references. External links, Gene Reviews NIHUW entry on familial juvenile hyperuricemic nephropathy type 2, OMIM entries on familial juvenile hyperuricemic nephropathy type 2, the MEREPS online database for peptidases and their inhibitors, A1.007, renin at the U.S. National Library of Medicine medical subject headings, renin at eMedicine Dictionary.